The title of this presentation is Protease Inhibition in mRNA Vaccine Development for COVID-19, an in-silico approach. To begin with a presentation overview, we'll be discussing a mathematical framework which is rooted in deep learning for studying activity of COVID inhibitors. And to begin this discussion, we'll motivate the importance of looking at protease inhibitors and how different types of inhibitors can interact with binding, and how this would help us determine the regions of the surface of the COVID-19 crystal structure and the actual where the actual receptor can interact within the structure when an infection for COVID-19 happens. And we'll be drawing upon information from referring to the protein data bank, which is one possible online repository which has information on the COVID-19 crystal structure. And this will allow us to obtain the PDB files, which are used for molecular docking simulations, which are comprised of a series of steps in a workflow that have been done before and described in another presentation, which I'll draw the attention of the viewer to. And we'll close out by establishing points of comparison between the predicted binding affinities of the deep learning framework and discuss what type of expectations for forming a comparison between the results produced from the deep framing molecular docking framework and another open source molecular docking framework to determine what are the drawbacks and different characteristics of each one of these algorithmic pipelines for being able to give us some structural biological knowledge. So the main paper reference that we're going to be using is something in quantitative biology under biomolecules from the archive from a couple of years ago. And what it looks at is unveiling the molecular mechanism of SARS-CoV-2 main protease inhibitor from 92 crystal structures. And what the authors in this paper look to do is that they look to determine how the possible binding pockets from the COVID-19 crystal structure can be deduced from a structural perspective. And they they predict that there are approximately 20 around 20 possible binding pockets, and then they perform simulations for quantifying the approximate binding affinity across each one of these binding pockets. And what's interesting about their work is that it leads to different possibilities and different avenues for establishing a comparison between another type of pipeline, which would make a similar use of the PDB structure, which is available from a online repository, but it trains a classifier from a machine learning algorithm a little bit differently, and in some cases much more differently, to form a prediction of what the predicted binding affinity will be. And the motivation for this is that for many different types of viruses, and even besides COVID or HIV, which has some sequence similarity to COVID, there are different characteristics and different structures of the infection when it happens, and actually how the infection and the genome of the virus is amplified and propagated to the host organism that the virus is infecting. And this is just to say that there are several other types of layers, which is provided from this BPS bioscience image on the side, which demonstrates how, in the case of COVID-19, the structure of the spike protein, as well as the membrane protein, the envelope protein, as well as the lipid membrane, are delivering the RNA, which is triggering the infection. And it relates to our interest in the title of this presentation as well, in protease inhibition, because a protease inhibitor is looking at this very mechanism through which the new novel genetic material from the virus's genome is being amplified and propagated to the host genome, depending upon how the infection occurs and the modality and severity of the initial infection. And as we're continuing our motivation, some public repositories that provide a lot of great information on being able to allow someone to look at some three-dimensional representation of this structure and being able to deduce what are the different structural characteristics amongst the approximately 20 binding pockets that are predicted from the deep learning framework that I had mentioned in the archive reference, we can look to the NCBI website, which is also related to the Protein Data Bank website, which are two really great references, which provide us with the PDB file, which is just basically a file that's used in the pre-processing step for either the deep learning framework and the other open source molecular docking framework alike, and how these two methodologies, even though they make use of some similar type of input data file from the government run online repositories that are publicly maintained, how there can be a lot of differences in later downstream pre-processing steps from this initial PDB file for forming some classification task on predicting the binding affinity. And this is to say that having a way to make use of an in silico experiment or another type of way to simulate the predicted binding affinity and to discriminate between which candidates or which compounds would be the best for different binding pockets of the COVID-19 structure is important, because it can be really difficult for producing different types of treatment at scale if there are some uncertainties about what's the difference in the performance of different candidates when forming some type of treatment. And this is just to show that amongst different types of phases, there were a lot of initial testing that was being done for vaccine candidates, but that even across the world, a lot of times there can be a bottleneck, and a lot of candidates or potential treatments were dropping out in the phase 2 and the phase 2/3 phases of the trial in comparison to phase 1 or phase 1 and 2 and phase 3. So it's just demonstrating that looking at ways to optimize and operationalize how well the pipeline would perform for sourcing together the raw materials that would be guided by an in silico prediction would also help improve the efficiency in which new candidates and new compounds can be tested. And now that we're in 2023, it doesn't seem as likely that maybe as many vaccines will be, or vaccine doses will be administered from the peak that was around 2020 and a little after, around the middle of 2022. So with that in mind, it still remains of interest, though, to determine if there were any possible mutations to any types of structures, either for COVID or any other potential structures for new viruses of interest that could be studied further, and how the algorithm could take into consideration different structural aspects of these proteins and these enzymes, and how they would react with one another when we're looking at them as as ligand and receptor pairs, and determining the approximate binding affinity between different pairs of compounds. And this is furthermore to show, in addition to the plot that was given in the last slide, is that we can also look at the worldwide distribution of vaccines, as well as boosters, which were administered, and there can be some type of lower proportion of vaccines which were administered for every group of 100 people in some areas of the world. And because of some sequence similarity between COVID and HIV, looking at some structures of HIV and how we would run an in silico experiment for looking at different ways of in which the HIV structure is from publicly deposited models like on NCBI or the Protein Data Bank can also be helpful. And it can be a good reference to have for a virus related to COVID, because both of them are RNA based. And this is what's given on the side, one reference from 2021, and it establishes similarities between HIV and SARS-CoV-2. So this group of several authors, they basically established parallels between HIV and COVID, SARS-CoV-2, due to the fact that they are both RNA-based. And it's also looking at even with the most recent vaccines, the use of the mRNA technology for messenger RNA for trying to anticipate ways to stop the virus from propagating its genetic material. And this is just to say that if we look in the public repository, as I alluded to in the previous slide, for our HIV structure, you can see that there are 23 possible models. And I didn't superimpose all 23 of the models together, but we can see that if we compare different types of models which are publicly deposited for HIV from the public databases, there can be some differences which would have a structural consequence depending upon the the structure of different loops relative to each other, as well as the geometry of these loops, which are given in the crystal structure for HIV. So this is just to say that when we're performing an in silico experiment, an advantage of having this approach and simulation method in hand is that it can allow for us to form different types of characterizations from a purely structural perspective without having to develop any types of assays or trying to perform any types of classifications from the binding affinity between different compounds from data, just trying to purely theoretically deduce what an approximate range of would be of the binding affinity and how we could look at different structures of compounds to determine which compound would perform better. And from a post on LinkedIn earlier in the year that I'd seen from GenScript, they provided a really great overview for different types of nanoparticles for drug delivery, which can kind of also come into play from the vaccine supply chain which I've mentioned and alluded to in the previous slide, as well as having to source together many different components and synthesizing and formulating these components for different types of treatments. And in this case, for drug delivery, this graphic really provides a really great overview because it demonstrates how you can either have a polymeric lipid-based or inorganic vehicle of delivery for these nanoparticles, which can be used for administrating treatments in different parts of the body. And you can just see also that from the middle column, from the lipid base, we can see that there are many different types of nanoparticles which can be employed, which depend upon different ways in which the drug molecule that is to be administered is configured within either a liposome or an emulsion, as two out of the four examples of the lipid-based nanoparticles. And in terms of protease inhibition and virus dynamics, this is important because if we make use of an in silico approach, it's really attempting to give some type of output which is reflecting the whole mechanism in which the maturation stage, as shown on this left-hand part of the slide, is undergone and it occurs. And the protease inhibitor is looking to play a role in this initial infection stage, because if there is no protease inhibitor, then there can't be any maturation stage. And then if there weren't any maturation stage, there would be no mechanism and no possibility for the virus to replicate its DNA and to amplify and propagate its genetic material to increase the severity of an infection. And this is just showing that from the middle image of the slide for HIV, we can see that in this CD4 cell, within the cell, there are HIV proteins and there's protease as well, but this is immature HIV, so there can be some conformational stages that these CD4 cells can undergo to transition from being an immature HIV cell to a mature HIV cell. So it's just demonstrating that in this budding stage, which is given in the middle image, HIV can transition from being immature to mature. And in doing so, when those CD4 cells reach maturity, it allows them to propagate different types of information and at different rates and speeds, which would determine the course and the severity and the duration of the infection. And in the final image of this slide, it's just demonstrating that the protease, which is belonging and neighboring the HIV proteins within the CD4 cells, can be stopped by protease inhibitors, which are not allowing the new HIV from becoming mature HIV. So because HIV does share some sequence similarities with COVID, as we had mentioned a couple of times and alluded to already, looking at this mechanism of protease inhibition for HIV and tying it to COVID in other types of possible mutations of the structure and determining how from the approximately 9 or 20 possible binding pockets that are predicted from the deep learning paper, how making use of protease inhibitors would prevent an infection from being propagated and amplified further. And so depending upon the vaccine formulation, several components of the supply chain can differ. As we can see from this reference from mdpi.com, they provide an overview of different types of purification and production of vaccines, and which was representative of the mRNA platform, which was used for Pfizer. And looking more closely at different types of these components of the supply chain unveils that there are usually a lot of stages which are involved with working some type of cells, whether they make use of a template or they look use of HEK cells or plant growth, or actually producing the virus. So each one of the stages has a production and a purification and finish stage, but all of these stages in terms of the operations and the supply management of the vaccine chain depend and differ depending upon the stage during which other purification and other linearization as preparation has to be done on plasmids. So it's just a variable stage for any one of these manufacturing protocols. And from the deep learning paper that we've been alluding to, the actual representation and very helpful visualization of the different protein binding pockets is demonstrated from one part of, or one perspective or one vantage of the SARS-CoV-2 crystal structure, and they annotate on the surface of the receptor of this protein the 10 or 12 possible binding pockets with other ones which are located on the other side of the receptor. So this is demonstrating that any one of these possible binding pockets exhibits a possible set of atomic coordinates that can be used in molecular docking simulations for determining what would the predicted binding affinity be, and how this can help inform and make faster the process of determining how protease inhibition also impacts virus dynamics, and how the virus, or really any virus for a receptor of interest, can spread. And from the possible binding pockets that were given, which were demonstrated on the previous slide, the paper from the deep learning framework makes use of looking at 92 ligands, which are distributed across 11 distinct binding sites. So in figure A, this figure demonstrates from the binding site index what is the distribution of the number of ligands per binding site, or per binding pocket or per binding site. And we can see that for the most part, the vast majority of the ligands fall in the first binding site index, which has approximately 70 of them. And then the other ones also have some candidates, but they're much more sparse. And from the 68, almost 70 possible ligands which are given in the first binding pocket, the deep learning framework allows for different predicted binding affinity energies in kilocalories per mole. And from the first paper reference on deep learning, the authors also provide another paper or another table in their paper, which gives an interaction analysis in the binding pockets of the top 4 complexes, which were formed from their MathDL or math deep learning models. And this is very much the background in a further framework that we're building upon from the motivation that was given earlier in the presentation, because we can establish frames of comparison between the performance of deep learning frameworks and also machine learning frameworks, and determining which input data can be provided to a deep learning framework that could provide it some type of performance advantage or operational advantage over just a machine learning framework, or whether vice versa, like, what would be the drawbacks from a machine learning framework, or if there were any advantages of using a machine learning framework. And this is just to say that for the top 4 complexes which are formed, they list the PDB IDs, which is coming into play from previous remarks that were made earlier in the presentation, because we can look up these PDB IDs to get started in the initial early steps of our workflow, and then running the pipeline across several potential pairs of ligands and receptors to determine what would be the corresponding binding affinity for every one of the complexes that are formed, and to perform a comparative analysis for determining which type of compounds would bind or perform better given a different binding pocket. And even whether we could perform on top of all of this, whether we could perform some type of unsupervised pipeline in which we don't feed any information a priori to the model about the actual distribution of the binding pockets, and determining whether the model could recover the atomic coordinates from each one of those binding pockets individually. So obviously this is a complicated task to pursue, because there are a lot of different types of structural considerations that we have to take into mind. To further demonstrate additional complexity and additional things that one would have to consider when we're looking at different types of residues and different types of surfaces of the COVID-19 structure. We also have to keep in mind that there are several different types of residues that are characteristic of SARS-CoV-2, in which typically appear, and the highest majority is GLI 143, which is approximately a little more than 70% of the total residues. So on the y-axis, we're plotting the frequency of different residues and how often, or what's the probability of selecting any one of these residues when we're looking at a portion of the SARS-CoV-2 structure. And in addition to considerations about what types of residues typically appear on the SARS-CoV-2 structure, as well as looking at the distribution of different binding pockets and observing that the majority of the binding pockets, almost 70 of them, appear in the first binding site. We can also look for violin plots, which is giving a visualization of the predictive binding affinity, which is given by the MathDL model when it's trained on different crystal structures, and looking at the consequences of this model and what would be the typical concentration of different binding affinities for these models when they're trained on the SARS-CoV-2 crystal structure. In particular, we can see that there can be different types of structural differences that can come into play depending upon whether there is a hydrogen bond or a covalent bond. And the 4 plots that they provide in this violin plot, it's demonstrating how the different aspects of the structure would impact the binding affinity energy measured in kilocalories per mole, from what's given either with no hydrogen bond, a hydrogen bond, a no covalent bond, or a covalent bond. So we can see that, typically speaking, the largest variation from the predicted binding affinities, which are produced by the MathDL model, occur when an H bond is present, because in the lower tail of the violin plot, we have a minus 9.03 kilocalories per mole. And in the top part of the violin plot, we have a minus 5.06. And these additional units demonstrate the mean, the average of the results of the predicted binding affinity from all of the MathDL models that are run from the candidate structures on the SARS-CoV-2 crystal structure. And to provide some further information on the format that this information that we've been discussing and comparing from results of the binding affinities is stored publicly, we can see that on the protein data bank, there is information that's given for the PDB entry. So in this case, we have 6Y2G. And this is important because it's some of the information that the authors made use of in their MathDL models for establishing which type of classification tasks they want to do from the structure and the actual the information that's provided in the protein data bank. And furthermore, on the protein data bank, there's also information that's provided in terms of a three-dimensional representation of a biological assembly. So this is demonstrating that this is looking at the Pfizer intravenous inhibitor. So it's the inhibitor that they had developed in their trials to characterize the amplification and the amplification stage of the virus. And the crystal structures provided in this image. And there are also many other types of experimental characterizations, and as well as determining, whether there are multiple models which are also deposited for this for 8DSU can also be determined like for any type of structure. And there are also other types of models which have different types of biological assemblies, which can be a little bit similar to the one which was given in the previous slide, but at the same time, they're not exactly the same, because there can be differences in either experimental characteristics or different type of assemblies, which can be useful to have and to also be able to run in silico experiments of for having different types of test cases in order. And this is all to say that there are even more similar crystal structures on the PDB which exist to the ones which were given in the previous slide. So in this case, I had just gone with a few of them just to try to test out things initially, rather than trying to distinguish between many types of differences between different types of crystal structures between these proteins. And to revisit the COVID crystal structure across all the inhibitors, we can see that if we look at a 180-degree rotation, a counterclockwise rotation of the crystal structure of COVID-19, which was given in a previous slide from the deep learning paper, we can see other types of another type of perspective of the binding pockets, which are given, which can also help provide us an idea or some intuition about any types of potential symmetry that this compound would have, which can be exploited and used in our either in a deep learning or a machine learning framework for classifying or predicting the binding affinity. And there's a second paper reference, which is also going to provide another helpful, other helpful characteristics of different types of possible databases, which can be used for looking at SARS-CoV-2 anti-SARS-CoV-2 drug candidates. And this ChemBL database helps also provide a different type of perspective on how the main coronavirus protease, which we have been alluding to and describing earlier in the processes of protease inhibition in HIV, how this can also be studied further from the structural type of framework that we have been describing. And in this paper reference, it provides some uses of Autodock Vina, which is the machine learning. It's a predominantly machine learning framework which we're going to be using, which is going to establish a point of comparison to demonstrate that under some circumstances, one could expect a deep learning and a machine learning framework to be able to perform very similar characteristics from predicting the binding affinity of different compounds. And in this case, we can see that Autodock Vina, like we're going to highlight, is a goal of making use of in silico experiments. Autodock Vina is helpful because it allows for us to look at different types of conformations within a binding pocket or within some active region once we specify the molecular coordinates, as well as being able to determine how different conformations can vary across this binding site or this active region or this binding pocket, depending upon the structures from the PDB files that we feed into the model initially. And as we had mentioned in another presentation on the channel, there are several different types of preprocessing steps which can be used for the in silico approach, in which we want to convert from a PDB to PDB QT files with the Python molecule viewer. So the Python molecule viewer is another really great open source add-on which one can use in addition to Autodock Vina, and it allows the person to look at different types of ways to interface molecules and determining how the binding pocket or the binding region of an Autodock Vina simulation would be able to look at different types of regions of a structure of a ligand and receptor. And in this presentation, which I had alluded to, which I had given on another thing on the channel, I had been looking at several different types of ligands and receptors in compounds in general from a CellTalk DB database, which is very much related to the spirit and the thought process of this presentation, because it's making use of looking at how different viruses propagate genetic material and how this infection process can be studied from conformational states which are obtained from molecular docking simulations. And the Python molecule viewer also has an open source documentation which can be downloaded from the Scripps Research Institute. And together with the PDB QT files, we implement the molecular docking simulations in Autodock Vina. So it's pretty much making use of incorporating the information which I had given in this presentation on the left, in which I was looking at some potential uses of looking at a public repository for other types of respiratory diseases or many other types of diseases in general, which have completely different receptor structures and different types of structural biology things that in considerations that we have to take into mind, and then actually looking to apply the flexibility of that framework for looking at the structure of the COVID-19 receptor, and in particular, the binding pockets, which I've been alluding to and discussing, and looking at how different binding pockets would perform within this within the Autodock Vina framework. And we can orient ourselves with Autodock Vina because they even have a lot of open source documentation and like video tutorials which demonstrate how to look at a molecular docking image using Vina on the Autodock tutorials with Pymol. So this is pretty much the tool that I also use, because it's a really great way to either be able to set the center of the grids of the atomic coordinates that you want to perform the simulation around, and then looking at different degrees of freedom or different number of points in the X, Y, and Z directions to determine how the box that you're using to capture that active site or that binding site would differ. And on the CellTalk DB GitHub, it actually shares in a lot of great potential and even future opportunities still in being able to apply or look at different ways to study the connection between in silico experiments, and being able to look at how molecular docking simulations would be able to perform for different types of random mutations or different types of mutations, directed mutations to the structure of to the crystal structure of different viruses. And this is just to say that even from the CellTalk-DB database, it's a completely different online database than the NCBI one or the protein data bank, but it still interfaces and still points to the protein data bank because there are several different types of data collection stages that they use in terms of annotation, as well as looking at different types of genes and performing additional pre-processing steps from this annotation information in determining how different pairs of potential ligands and receptors could be constructed and could be formed for actually studying different types of viruses or diseases. And so, yeah, this is just an overview of their website and the ability to look at different ligand receptor interaction pairs. And besides this, though, we also have to generate homology models, which is going to be very important because it's going to be one of the input files which we're going to feed into our model in Autodock Vina for performing a classification task on what would we predict the binding affinity to be. And this is coming into play because when you run a homology model on the Swiss homology modeler, typically there is a summary tab which provides a higher level overview of what types of characteristics of the protein data bank structure and file we would expect to have from just a sequence that we feed into the homology modeler, but as well as the fact that there can be some templates and models, 16 and 4 in this case, which each have different types of characteristics, namely including the oligo state, the GMQE score, which is a perceptron score, which is relating to the classification error, and the sequence identity. And we can see that some candidates from the list of possible homology models can be pretty complicated, which can demonstrate the utility. And again, as I mentioned earlier, the flexibility of this approach in being able to look at different types of viruses and how their dynamics and methods of propagating infections would differ, and how we can look to different conformational representations and different binding affinity approximations that we gather to inform our thought process and knowledge of these different viruses. And the implementation steps for Autodock Vina include that we have to look at each different type of possible conformation. So we can see that even from some other types of simulations from Autodock Vina that I've ran, we can see that there can be different types of possible conformations. For instance, we can either have 9 or 6 possible conformations produced, and that's given in the first column, in the left handmost column of each one of the simulations, and in the second leftmost column, we also have the affinity that's predicted in kilocalories per mole. And the conclusions from the CellTalk-DB sequences and how it's going to connect to the studies and application of the Autodock Vina framework and molecular docking framework in general to the SARS-CoV-2 receptor is that it motivated current interest in academia and industry for simulating activity between ligands and receptors, because there are several different types of ligands or receptors which are under consideration under many types of groups in either in industry or academia, which are interested in either improving or optimizing performance or improving binding affinity. And after further discussion beyond the motivation, we introduced CellTalk-DB. So CellTalk-DB, it's allowing for us to look at different types of ways to gather information on potential ligands and receptors, and how these groups of ligands and receptors are reflecting different characteristics of the viruses that are looking to infect some host organism. And as a public resource for academic use, CellTalk-DB provides many combinatorial targets that we can investigate, including human and mouse receptors and corresponding ligands. In building upon the CellTalk-DB resources, we describe the results generated by Autodock Vina for 2 ligand receptor pairs, and there are several other compounds of interest whose homology models I have in some resource on my GitHub. But this is demonstrating the type of framework that we can use to and apply for the SARS-CoV-2 receptor. So we can set up the molecular docking simulations and the coordinates. So we have to directly run in the terminal or some type of Linux environment, the type of search procedure that we want to do, so that we can look at the type of receptor, the type of ligand, in specifying the center in X, Y, and Z dimensions of the molecular docking simulation, as well as the number of points or the size of each one of the dimensions in the X, Y, and Z dimensions. And in this case, we see that we looked at the receptor for 7BQY, and the ligand was 5RW2. And what we've been wanting to do is to form a comparative type of analysis for interpreting results from conformations that are produced in the PDB-QT output. And then what we want to do with this molecular docking in silico is specify the molecular coordinates for determining if there would be any point of comparison with the deep learning framework, and in particular, whether the deep learning framework would have any point of comparison. And unfortunately, in that part, I'm still running the simulations and being able to finish them up, but I just ran into some potential difficulties for now with my workstation and being able to finish up the simulation in this last slide. But I'll probably update this the link in this bio within a couple of months to actually describe what would the results be. But even though the slide is blank for now, I think that we can look at different types of possible performance comparisons between the MathDL framework and the machine learning framework, and that could either be related to there could be some type of binding pocket which is captured by the MathDL framework, which is not captured by the machine learning framework, which would not be good and not be very not reflect very positively on the performance of the machine learning framework. Or there could also be a potential difference in the predicted binding affinities from any one of the compounds which are given from each one of the almost 20 possible binding pockets. So the conclusions is that we introduce a mathematical framework which is rooted in deep learning from the archive preprint, and we describe properties of the surface of the COVID-19 receptor on how one can approach in silico molecular docking experiments amongst different binding pockets. And for a couple of the binding pockets about which the majority of the activity is predicted to take place, we provided visual renderings of the complex formed after the binding interaction from the PDB-QT files produced by Autodock Vina, and additional work and future updates will be provided for studying the activity and structure of different complexes from new COVID variants using Autodock Vina. And thanks for listening.